At this point, we've now taken a look at the structure and function and evolution of the different vessels in the circulatory system. In this web lecture, we're going to take a look at the evolution of the heart itself and the structure of the heart. So we've already seen the structure of a typical fish heart, this kind of flow-through four-chambered heart. And we're working our way toward this very much more complex, still unidirectional flow-through heart, but with this complex pattern of blood flow involving lungs returning to the heart, being pumped again. And so what we want to see in this web lecture is how do we get there from here? So let's start just by reviewing the different chambers of the fish heart. So beginning with the sinus venosus, which remember is sort of a collection chamber as blood enters the heart, leading into the atrium, which is lightly muscularized. It's going to be responsible for making sure that the ventricle, the next chamber, fills adequately with blood so that when that main muscular pump, the ventricle squeezes on it, it's going to give it maximal pressure because it's filled to maximum capacity. The ventricle is then going to send the blood out through the conus arteriosus, which again is a heavily muscularized vessel whose job is to resist those pulses of pressure from the ventricle and even out the blood flow as it is transmitted out to the ventral aorta and then the gills. So they're not experiencing these very dramatic pulses of pressure from the ventricle. They're experiencing a nice even flow. So we've got this basically tube-shaped heart with a little bit of a curvature to it with unidirectional flow going in and then out in a single direction all the time. So how does this change when we start to get to something like an amphibian heart with the two-circuit circulation being diverted out to the lungs and then back to the heart? So we can think of this heart as just kind of getting pulled together in a couple ways to get that single heart structure that we're used to. So if we think of the atrium being kind of pulled up toward that conus arteriosus and the sinus venosus kind of getting pulled toward the ventricle, this is kind of what we start to see in the amphibian heart. So remember, in the amphibian heart, we see the first real septation of the heart. So there's going to be an interatrial septum separating the right and left atrium. And then if we think of this folding, we can again think of this atrium being kind of folded over, coming close to the conus arteriosus, the ventral kind of being squeezed up close to the sinus venosus into this kind of folded structure with the ventricle at the bottom, the two atria up above it, the sinus venosus as still a separate chamber off to the side. So in amphibians, we're going to retain the sinus venosus as a separate chamber. You can see it here. And then again, we get complete septation between the right and left atria. So there's that interatrial septum separating the two. So if we look at the blood flow through an amphibian heart, we no longer have this simple unidirectional flow with no mixing of oxygenated and deoxygenated blood. So if we imagine the deoxygenated blood coming in here, this is the entrance of the sinus venosus into the atrium. The atrium is going to expand while the ventricle contracts, so this blood is going to be forced into this expanding right atrium. We're kind of looking at this with the conus arteriosus sort of sitting in front of the atrium. So imagine this blue arrow kind of going back behind the conus. So this is a section of the right atrium here. This is a section of the right atrium. And the blood is filling that chamber. Then the atrium is going to contract while the ventricle expands, drawing the blood from the right atrium down into the ventricle. Then when the ventricle contracts, that's going to force the blood from the ventricle into the conus arteriosus. Now the conus arteriosus has a spiral valve that causes blood to be preferentially directed toward the particular section of these branches of the conus arteriosus known as the truncus arteriosus. It's going to cause that blood to be diverted into the portion of that that's going to take it to the pulmonary circuit. In the case of the frog heart, that's going to be the pulmocutaneous artery, taking the blood both to the lungs and also out to the skin to pick up oxygen. When oxygenated blood comes back from the lungs and the skin, it's going to enter the left atrium through the pulmonary vein. Again, this is going to be while the atria are expanding, the ventricle is contracting. Then the atrium contracts, the ventricle expands, driving the blood from the left atrium into the single ventricle. And again, this valve is going to mostly direct this blood that's coming over here from the left side toward these other two segments of the truncus arteriosus to the carotid and systemic circuits. And so this seems like a very sloppy system. It seems like there's a lot of potential for mixing of oxygenated and deoxygenated blood, which in fact there is, but studies have shown that this is remarkably effective at keeping the blood separated. Most of the deoxygenated blood, it turns out, does in fact head toward the lungs and skin using just these little flaps and valves, and most of the oxygenated blood does in fact head toward the brain and the systemic circuit. So there's all this potential for mixing, but in practice it actually works pretty well. Now let's take a look at the non-archosauriid reptiles, the lepidosaurs and turtles. So they retain the sinus venosus. It's very much reduced, and it's associated with the anterior and posterior vena cava and has this entry into the right atrium. In these non-archosaurian reptiles, we start to see some septation in the ventricle, but it's only partial. There's a structure called the interventricular canal, which we will think about in great detail in class, that allows some mixing of oxygenated and deoxygenated blood within that section. It's sort of a conduit between the two sides of the ventricle. The conus arteriosus at this point becomes incorporated into the walls of the ventricle, so it no longer exists as its own individual structure. And as we saw when we were looking at the evolution of the aortic arches, we now have a ventral aorta that is completely divided into three separate vessels. In crocodilians, now the sinus venosus no longer exists as its own separate chamber. It becomes incorporated into the wall of the right atrium. And now in crocodilians we see full septation of the right and left ventricle. So there's a complete wall here separating the right and left ventricle. And the conus arteriosus is now incorporated into the walls of the left ventricle, so this more muscular side of the heart. But we still retain this passageway between the oxygenated and deoxygenated blood, the structure called the foramen of Panizza, that connects together the left and right systemic arch. So again, we'll talk about the function of this in more detail in class, but this is the only place in the crocodilian heart where oxygenated and deoxygenated blood can encounter each other and mix somewhat. In birds, this foramen of Panizza is lost, and we have completely separated pulmonary and systemic circuits with no possibility of mixing oxygenated and deoxygenated blood. This is also the case in the mammalian heart. So in the mammalian heart also, the sinus venosus is incorporated into the wall of the right atrium, and it becomes the structure called the sinoatrial node. This is the pacemaker that maintains the rhythmic beating of the heart and the coordination of the heartbeat between the different chambers. So let's see how this works. So blood enters into the right atrium through the superior and inferior vena cava. In the case of the human heart, we call it the anterior and posterior vena cava, or precava and postcava in the case of other non-human mammals. As the right atrium contracts and the ventricle relaxes, blood is forced from the right atrium into the right ventricle through the right atrioventricular valve, also known as the tricuspid valve. When the ventricle contracts and the atrium relaxes, this tricuspid valve is pushed shut by the blood pressure acting on it, forcing the blood up into the pulmonary trunk through a valve called the pulmonary semilunar valve. So this is a flap of tissue with little openings on it. When the blood is pushed through, it opens, but when blood is pushing back against them, it snaps shut to prevent retrograde or reverse direction flow, keeping everything flowing in the right direction through these semilunar valves. So from there, the blood is going to go out to the lungs through the pulmonary arteries to become oxygenated. Then they'll return to the heart through the pulmonary veins into the left atrium. So again, left atrium expands, left ventricle contracts, pulling blood from the pulmonary veins into the left atrium. Then the left atrium is going to contract, the left ventricle is going to expand, pushing blood from the left atrium into the left ventricle. So the left ventricle is going to be the most heavily muscled part of the heart. This is where the very most pressure is going to be applied to the blood, because this is the pressure that's going to need to drive the blood all the way through this whole systemic circuit. And in a large body size, it's going to take a lot of pressure. So this is going to be very thick-walled, very heavily muscularized in the left ventricle. So as the blood moves from the left atrium into the left ventricle, it's going to pass through the left atrioventricular valve, also known as the bicuspid valve. And these flaps of tissue are going to shut when the ventricle contracts to prevent backflow from the ventricle back into the atrium. So when the ventricle contracts, it's going to force the blood out into the arch of the aorta to be delivered out to all of the body tissues. And as it moves into the aorta, it's going to pass through, which you can just barely see right here, another little flap of tissue, the aortic semilunar valves. It's going to look similar to these pulmonary semilunar valves. And again, these valves are going to prevent any backflow or retrograde flow through this heart. Similar to the fish heart, we now see unidirectional flow. Everything just goes through each chamber one time. We don't have any mixing of oxygenated and deoxygenated blood as we saw in the amphibian heart and the non-archosaurian reptilian hearts. We've got a complete separation between the pulmonary and the systemic circuit in the hearts of mammals and birds. So, the end goal of this whole section on the circulatory system, what I want you to be able to do is trace the pathway of blood flow from any structure in any of the vertebrates whose circulatory system we've described to any other point going through the heart. So you'll be given a task sort of like this. Imagine you're a red blood cell in the anterior or superior mesenteric artery of a cat. Using the names of structures and arrows, trace the path you would take to get to a capillary bed in its left forelimb. Remember, if you're going from an artery in one region to a capillary bed in another region, this is always going to carry you back through the heart to get to that other location. So, including all the major vessels, locations of capillary beds, and chambers of the heart. You may ignore the valves in this case. And I've given you a couple of steps leading to the anterior superior mesenteric artery to get you started. So, in this example, we're starting in the arch of the aorta. We're going down to the dorsal aorta. In mammals, it's generally just called the aorta since there's no ventral aorta, and into the anterior mesenteric artery. So from there, we're going to go to a capillary bed in the small intestine. Remember that the anterior mesenteric artery is that unpaired vessel that serves the midgut, including the small intestine. From there, it's going to go to the anterior mesenteric vein. So remember, unless you're told otherwise, these veins are going to just follow the arteries and will be named similarly. So from the superior mesenteric vein, remember, every drop of blood that enters one of these unpaired arteries going to the digestive tract always going to go through the hepatic portal system. So it's going to go from there into the hepatic portal vein. Remember, a portal system goes artery, capillary bed, vein, capillary bed, then to another vein. So the other capillary bed we're talking about in the hepatic portal system is a capillary bed in the liver. From there to the hepatic vein. The hepatic vein is going to join up with the posterior vena cava, which is going to lead into the right atrium, into the right ventricle, up into the pulmonary artery, into a capillary bed in the lungs, into the pulmonary vein, into the left atrium, left ventricle, and then the aorta. From there, it's going to go into the left subclavian artery. You could include also in here the left brachial artery. Remember, the subclavian arteries become the brachial arteries once they get out into the limbs, and then from there to a capillary bed in the left forelimb. So practice doing this. Think of any spot in the body of any of these groups of vertebrates that we've talked about, and make sure that you can trace the blood flow from any place in the body through the heart and back out to any other place in the body.